Hello, this is Dr. Stephen Schenzel, Professor of Community Medicine and Healthcare in the Department of Community Medicine. And I'm here to provide you a brief review of a project focused on alcohol and ART adherence among persons living with HIV in Mumbai, and those people are on ART medication, antiretroviral treatment. The project was funded by the U.S. Institutes of Health, particularly the National Institute of Alcohol Abuse and Alcoholism, and includes a number of collaborative partners that you see on the screen. I won't go through it, but I might mention some of them as I'm going through. Well, we focused our attention on 5 ART centers located in major hospitals in Mumbai and in the Mumbai area, and also very much involved with the National AIDS Control Organization and the counterparts at the state and Mumbai municipal level. The issue of alcohol and HIV has been well documented. Alcohol consumption and excessive alcohol consumption increases HIV risk through risky sexual practices. It negatively affects immunological function in persons already having HIV. It contributes to poor ART adherence. We'll hear some information about that later on in this presentation. It increases the impact of the disease on the human body through, as measured by CD4 cell count that's lower for those who consume alcohol, and a higher viral load, contributing to the potential for greater amounts of transmission to HIV-negative partners and fewer years of survival. And finally, all the issues associated with alcohol, even for non-HIV people, are exacerbated by those whose immunological functions are lower and produces pulmonary and liver disease, cardiovascular disease, mental health issues, and central nervous system disorders. Our theoretical model is a little complicated, but indicates that there are reading from left to right subgroups based on sociodemographic factors that might include ethnicity, income, geographic location, nature of housing. A second panel that deals with biopsychosocial features, including the nature of the family structure and the level of poverty, as indicated by food insecurity, social support networks, as well as peer structures that would support the individual living with HIV. We'll call them PLHIV. Of course, alcohol use in this project, psychological well-being, and the degree of comorbidities that might be present that the individual has to cope with as well. In the third panel, it involves the nature of services being provided and the degree of response of those services, including the ART center services themselves, other healthcare services that might be in the hospital or in other locations, or even non-allopathic healthcare, and HIV Positive People's Network. Each state has a Positive People's Network, and Maharashtra, the state in which Mumbai is in, has a especially active one that's played a very significant role in our project. Finally, we're looking at the outcomes of ART adherence. The level of disease status is measured by viral load and CD4, and risky sexual behavior. In this case, particularly the potential for transmission to wives for those who are married. And then the results to be generalized to PLHIV in ART treatment throughout Maharashtra in terms of a number of different factors. The intervention design is also a bit complicated, but we think very interesting. We're going to provide for PLHIV individual level counseling, a one-on-one counseling between the ART counselors and some of our project counselors. A second intervention would focus on groups that would be facilitated by a network of positive people plus the ART coordinators for mutual problem solving. And at the community level, perhaps some of the most innovative is to organize men for collective action to address internal stigma, their own stigma about having HIV, but more importantly, the external stigma of their denial of human rights in terms of entitlements, in terms of general reactions of the public or service personnel. And so this is a collective action advocacy effort, particularly facilitated by the network of positive people. We have recruited 940 individuals, 188 at each of the centers. And Center 1 would get individual counseling. Center 2, group intervention, and Center 3, community intervention, with 2 controls. Results I'll give you in a little bit describe the outcomes of Cycle 1. We're just in the middle of, or just wrapping up Cycle 2 at the moment, and anticipate Cycle 3 over the course of the next year. So Center 1 that got individual will go on to Cycle 2 and get group. So they'll have individual plus group. And then in Cycle 3, they'll have community, and so on. The second center will have group, community, and individual. The third center will have community, individual, and group. And that will allow us to understand this is a stack design involving both the sequencing and packaging, allowing us to understand what intervention was most effective and what sequence of interventions were most effective. A slight alteration is in Cycle 3, we have designed a full package of individual, group, and community interventions, and those will be just being organized and implemented. So that will be implemented in Cycle 3, and the fourth center will be control in the first cycle, control in the second cycle, and individual and group and community in the third, and the fifth center stays as control. Our steps in project development involved engagement of the national, state, and Mumbai district AIDS governmental organizations, both as a process of allowing the project to be implemented, providing approval, but also to engage key individuals in the project itself. So this was we very much have developed partners at each of these different levels, and they become very active in the project. The next level was developing relationships and collaboration with AR Center PIs. Those who were heading the ART centers became part of the research project as well. The third area was engagement with the Positive People's Network, who have supported the project and who have identified facilitators for both group and community intervention. We moved on from there to do qualitative assessment through observation of ART centers, matching the guidelines provided by the National AIDS Control Organization to the actual services that are being provided. From there, we moved on to key informant interviews with providers, those individuals with special expertise, able to represent what's going on, what are the challenges, what are the issues. And one of the challenges is that the centers were developed for 500 people, now have 2,500 to 3,000 people providing services. We then did 50 in-depth interviews, qualitative in-depth interviews, exploring various life circumstances and life history of the individuals with HIV who were on ART and also consumed alcohol. And finally, the administration of a baseline survey to the 940 PL-HIV we recruited. The use of qualitative data is important, and I'll cover that in the next component that I provide for this coming session. This is an example of some of the things we've learned from individuals and some of the quotes and that were coded to support these results. We found that drinking with others was drinking with friends, and particularly led to situations of going to a red light district or with commercial sex workers, and men identified themselves as having been the place where they contracted HIV. So the qualitative data provides us a great deal of discovery, whether it's patterns of alcohol use, how individuals contracted HIV, what are the dynamics in the family in dealing with PL-HIV and dealing with HIV and AIDS, and a number of other issues. As we discovered these elements and could build them in both to the survey instrument and to the nature of the intervention itself, as well as providing data that we've used for publications. Our quantitative variables focused on outcome and predictive variables. We used audit. Dr. Baber has been crucial in the development of the use of both audit and assist. We used audit. We used the timeline follow-back, which is a 30-day calendar in which people recall the specific days in which they engaged in alcohol behavior. And we expanded and modified that. A new publication coming out in AIDS and Behavior describes the results of looking at not only alcohol use and how it distributes among the last 30 days, but as well sexual risk behavior and adherence behavior, and looking at the interrelationship. We'll see that in a bit. Biological indicators, as I mentioned, a CD4 count and viral load, sexual behavior, frequency of sex with partners, timeline follow-back that told us when they were having sex with a partner and whether they were using a condom, and the percentage of the ratio of number of times of sex with wives and the percentage of condom use in those times. In addition, we've looked at the depression and tension. Tension is another scale we've developed from our ethnographic work that describes a particular component of the way people address and conceptualize anxiety and stress factors. So this is a term that's been translated into Hindi. We've looked at depression for the CESD and both internal and external stigma. Both scale, the Berger scale for external stigma and an ethnographically derived internal stigma scale. And finally, symptoms associated with being on ART. From the timeline follow-back, we note that when we have these risky elements on the same 30-day timeline follow-back, we're able to identify that participants who drank on a particular day were less likely to adhere to medication on that same day. And participants who drank were less likely to adhere to medication the day after they drank. than participants who didn't drink. Again, participants who were more likely to have unprotected sex on the same day that they drank. So drinking was associated with unprotected sex on the same day, and but were less likely to have unprotected sex on the day after they drank. The results of our treatment and control group, what we did in our initial analysis was cluster all of the experimental, the samples from the 3 centers that were experimental with the 2 centers that were control. And what we see here is that there's a significant reduction among the treatment and experimental group in the audit score, a reduction in reasons for drinking, and a significant reduction in a significant increase in four-day adherence to ART medication. For tension and depression, repeated measures indicated that tension was reduced in the treatment group and significantly reduced in the treatment group, as was depression, but as compared to the control group. The reduction in an external and self-stigma was also clear and highly significant, showing that both external stigma and self-stigma was significantly reduced as opposed to controls. To summarize this component, we then looked at the efficacy of the individual, group, and community interventions. And what you see here is a quick summary table that indicates that the individual counseling had an impact on, a positive impact on reasons for drinking. That is, there were less reasons for drinking. There was a reduction in tension and a reduction in depression. The community group had a reduction in reasons for drinking, self-stigma and external stigma. And the group intervention seemed to be the most effective, having an impact on all of these outcome variables. Let me make the point, though, that the individual counseling, it seems to make sense that the individual counseling would focus on internal psychosocial issues, that the community intervention would focus on their objectives of reducing both the stigma of individuals participating in the groups and would affect external stigma. And the group intervention seems to be successful on all of these elements. The sensitivity of the evaluation to the individual and to the types of interventions is a positive development. But also, remember, we're going to sequence these, so we yet don't know how individual will affect, how in the first center, how individual plus group will affect the outcome, or individual, group, and community affect the outcome. And that will await the rest of our work. So that's our design, some brief analysis, and I hope this has been a useful example in the context of the clinical work. Thank you.